Welcome to the July Tech Talk from Nuance BioCryo this time. My name is Rainer Blair. I'm the manager of the BioCryo core at Nuance. So the title of my talk is Automated Electron Microscopy Sample Preparation with the ASP-1000. So ASP stands for Automated Sample Processor, and the ASP-1000 is an instrument from Microscopy Innovations. So before we talk about automation, I want to go over the steps that are required to do sample preparation for TEM. So we first have chemical fixation. That means we cross-link the proteins in order to preserve the ultrastructure. We have different fixatives for that and different buffers depending on the purpose of the study. The fixative is rinsed out. And the next step is called post-fixation. It means we incubate the sample with osmium tetroxide. This reacts with lipids and in this way stabilizes membranes and also adds contrast to those. The next step after osmium tetroxide is rinsed out again, we incubate the sample with uranyl acetate. And the positively charged uranyl ions attach to negatively charged sites in the sample, and this is adding contrast in general. The next step is then, again, we rinse out the uranyl acetate and then we start dehydration. Dehydration means we replace the water in the sample with a solvent, usually ethanol or acetone or acetonitrile. And we do this by incubating the sample with an ascending series of this solvent. Once all the water molecules are removed and replaced by the solvent, we can start with resin infiltration. So we have different resins. Again, it depends on the purpose of the study. And there are different conditions that need to be fulfilled for resin to be applicable for EM. It's listed here. I will not go into details about that, but what we do for infiltration is, again, we use increasing concentrations of the resin monomers to infiltrate the sample. And once the sample is fully infiltrated, we can polymerize the sample and we end up with plastic blocks with samples in a firm matrix that allows us to make sections with ultramicrotomy for all the different thicknesses we need, depending on our application. So this is true for TEM. For SEM, we have basically the same beginning steps. We do fixation, post-fixation, dehydration, but then we would not infiltrate with resin, but do the critical point drying, mount this dry sample, coat it, and then it would be ready for SEM. So when we look at the time that we have to invest to complete all these steps, so each fixation, post-fixation, staining, dehydration takes at least, for a very simple standard protocol, 1.5 hours each. And this means it keeps us busy one day in the lab. We must also see that resin infiltration usually includes a step that goes overnight. And in the next morning, we still have to do some liquid exchanges with the resin before it's ready to be transferred into molds and can be polymerized. So this is another half day that we have to, time that we have to invest. So, and then the sample, of course, gets polymerized and we can proceed as usual. Also, when we look at this, we can see that at least 20 or over 20 liquid exchanges are required for each sample just until the end of dehydration. Depending on the protocol, it could be even more steps. And this is per single sample. So if we would have, let's say, 4 samples, which we usually do, that would be already 100 liquid exchanges that we have to do manually in the lab. And this, it's of course also time that is required. So, and this is why it would be great if we could do some of these things in an automated way. And that is why we have this automated sample processor from Microscopy Innovations. And that allows us to do things faster with different protocols that can be tailored for each sample that we need. So, and once we have, we use this instrument, we don't need to do the rinses after fixation anymore. We can do fixation itself too with the instrument, but usually samples are already in fixative when we start mounting them for the processing. So we don't need to do the post-fixation, the unblock stain, the dehydration, or the resin infiltration. All we have to do is place the samples in the capsule, start the protocol, run the protocol, and once the sample is ready, we just need to put it in the oven for polymerization. And then, of course, we still have to do ultramicrotomy and our studies. So the instrument we are using for this is the ASP. It's basically a liquid handling robot with a pipette head or pipette arm that transfers the sample through multi-titer plates from one solution to the other and performs some action at each position. And this whole system is designed for two types of capsules. So we have the, here on top, we have the MPrep S capsules. S stands for sample, in which the sample is trapped at the very tip of those capsules. Or we have the MPrep G capsules, which can hold 2 grids each. Those capsules are fixed onto the pipette arm and then transferred or transported or moved through all the necessary protocol steps. So how does the workflow look like? So first of all, we have to set up a protocol for the robot. Then we have to prepare the plates with all the solutions that we will use. The nice thing here is, of course, the protocol can be set up days or weeks or months before the experiments, and the plates can also be prepared before the experiments. So we don't have to do that exactly on the day when we have the samples. We can seal those plates and store them until usage. The next step then on the day of the experiment would be to mount the samples. That means we put them into those capsules. They are kept in place with a little screen. And once the mounting is done, we can attach the capsules to the pipette arm. And then we just run the protocol and we can walk away and come back when the samples are infiltrated and ready for polymerization. So, and then all we have to do is, of course, retrieve the polymerized samples for making sections or doing other examinations. So the first step, setting up the protocol, is done with the so-called dashboard control. That's the software we are using. And there are 2 basic modes that we have with this software. One is the operator mode. That is what usually the user would encounter. So all the user has to do when the protocol has been established well before is to load the correct protocol and to make sure that the correct tray file is loaded. You can then see here in the left the layout. So we have space for 6 microtiter plate that can be 96 well plates or 12 trough plates, depending on the step and if you have different treatments for samples. And the user then just has to start the protocol. There are also some dialog boxes or interactive boxes. So first the software wants to confirm that the correct protocol is loaded. If that is the case, it will proceed initializing the valves and the pump. And then it will ask to attach the samples to the pipette head. And once that has been done, the user has to press enter, and then the protocol can start. There's also a possibility to put in pauses into the software so that the user can interact or add solutions if they are extremely volatile, or if there are any circumstances that require that the user needs to interfere with the process, which usually is not really the case. We also have information that the program gives us. So it tells us for each single step how long the step still will need for time. It will tell us when the step exactly will be done. It tells us also how long the whole protocol will still need, and also the exact point of time when the protocol is done. So you know when you can come back, for example, to retrieve the samples. Also on the map is always indicated at which position in the protocol we are at the moment. So this is the operator mode. The other mode, of course, is the developer mode, and that is the mode that we use to actually set up the protocols. So this is a very intuitive software. So we have here a pull-down menu for commands. The mostly used command is move, aspirate, dispense, which tells the pipette arm where to go exactly with the sample, which plate into which column, and to perform which actions. So here we see plate 3, column 2, which is also indicated here. And what we can do here is we can adjust all those parameters. We can adjust the speed, how fast the fluid is aspirated into the sample capsule. We can also adjust the volume that is being used. And we can adjust if there is a hold after each of these movements, which can be very useful for samples with higher viscosity. And we also can, sorry about that, can of course adjust how many repeats we will have. And so this is makes it really a very effective system. A typical protocol that we have used not too long ago here is this one here. So we can see most samples, most commands are MAD, move, aspirate, dispense, always telling to which plate and which column to go. And the differences are usually not in the volume. We are always using about 100 microliters. I think the maximum volume we could use is 150 microliters for a single capsule. But we see we have differences here in the repeats. So for the rinses have less repeats than incubation times. And we also have differences in speed. Yeah, where can I see that here? So we have speed here. So we can see lower numbers of speed means it's faster. And when we go into those resin steps, we can see that we use much lower speed numbers because of the viscosity of the resin. So this is the first step, setting up the protocol. The next step is to prepare the plates. And we are using polypropylene plates because they are compatible with solvents and also with the resins that we are using. And we have different types. So we have 196 well plates in general, can be plates with round wells, with square wells, with round bottoms, with flat bottoms. Or if all samples have the same treatment, we can use those trough plates. So all 8 capsules will dive into the same trough and liquids will be aspirated and dispensed. And here we can see again what I've mentioned before. We can make those plates in advance and we can seal those plates and store them so they are ready when the day of the experiment comes. Yeah, so volumes that are required, usually 3 to 4 milliliters per trough here, and about 100 to 200 microliters per well. So it's not too much volume that is actually required. So the next step, once the plates are prepared, is to load the samples, to mount the samples into the capsules. So we can basically use any sample that does not disintegrate during the process. So we can use tissue pieces, we can use hydrogel pieces, or pieces of anything that can be cut into small enough cubes that we can place in the capsule. If we have cell culture, for example, we harvest the cells, we make a pellet, and we can then resuspend this pellet in a very small volume of low melting point agarose. And this agarose it hardens at room temperature, and we can also cut it into small cubes for placing in our capsules. So here on the right, we see the whole system. So we have the main unit, the robot with the pipette arm. We have space here for 6 multi-titer plates. We have the pump system that agitates the liquids. And here is the laptop that controls the whole thing. So again, here in more detail, the pipette head with the attached sample capsules. So mounting the samples into the capsules can be a bit tricky, and that is why we have this nice workstation to do that. So this has wells that can be filled with buffer where we can put the capsules in with the samples so the samples do not dry out. We have dissection wells, that means those wells can be filled with buffer, and in these wells we can dissect the samples into smaller pieces. We have an insertion tool that can be used to place the sample in the capsule. And when we work with that, it looks actually like this. So here we can see we have several capsules here sitting in these wells. We have here wells filled, dissection wells filled with buffer and specimens. We have the little screens that keep the samples in place. And once samples are dissected, I can show you a little sequence how we mount the screen. So first of all, we have to place a screen here in this insertion tool. So we can do that either with using a capsule as we have seen, but we can do that also with a tweezer or by hand. Once that is done, we can use a clamp mechanism that is good for samples that are sturdy enough and that need to be kept in a certain orientation. But usually, if we don't need to do that, we would just place our sample piece directly here on this screen. Once the sample is placed, we can again come with our capsule, turn it around, and then the sample is mounted in the capsule and kept in place with this little screen. Okay. So, and here we can see the robot in the midst of the process. Again, we can see all the sample capsules are submerged into a bell here in a 12 trough plate, and here we can see this robot actually in action. So you can see the liquid here, that is already a resin step going up and down. And we can adjust, as we had said before, we can adjust the volume, we can adjust the speed, we can adjust if there is a stop in between. And this makes it clear that this is a very efficient way to exchange liquids in a sample. It's certainly way more effective than just using placing a sample in a vial in a solution and then let just diffusion do its work. So here we can see the capsule again at higher magnification. So we see our tissue piece here. We can see the screen that holds it in place. The capsule itself has 37 openings. So we have quite a good stream of liquids going up and down when we aspirate and dispense the sample. And again, so we can control all those settings like how many repeats, which speed we use, which volume we use, and if there should be holes after each step. And this is really important to adjust for the viscosity of media. There's also the possibility to stack capsules. So if we have really quite a lot of sample to process at once, and for stacked capsules, we have another command which is called mix. And that means once the capsules are filled, we are just moving the whole liquid column up and down also for a defined volume of liquid, of course, until the media is aspirated, is dispensed again, and we are ready for the next aspiration cycle. So here we see all those components again. We see those sample capsules with the screens. So we have a tool to place the screens. We can use the workstation to place the screens, or we can even use a tweezer. So here we see the grid capsules. So each capsule can hold 2 of the grids. And here we see the whole unit again, pump, controllers, the robot itself, and the laptop with the software. So the last step after each run is usually that all the capsules are placed in a silicon plate. This silicon plate seals the bottom. And since in the last step we always have the resin, so the sample sits then in the resin. And when the user comes, all he has to do is to disconnect those capsules from the pipette head, and then he can move the plate with the capsules directly into the oven for polymerization. We also have sample holders for critical point drying of capsules and for some trial applications. So here we also have a multi-pipetter that can be very useful for preparing those 96 well plates, of course. And yeah, I've shown you we can use different types of plates, like trough plates or well plates. Okay, here we see this workflow again. So we have the protocol set up, we prepare the plates. That can all be done before the experiment. We mount the samples on the head. We run the protocol, and then we detach the capsules and just transfer this silicon plate into the oven, polymerize the sample, and then we can retrieve the polymerized capsules and do the trimming and sectioning direct from the capsule block. So here we see an example for a clinical application where there are many biopsies need to be processed at once. So here we have those stacked capsules. We place 8 samples in each capsules. We have 2 capsules per board. And with 8 capsules, that means then we can process 128 samples at once. So this is really a very effective system. And we can also have very short protocols. In this case, samples were not polymerized in the capsules, but were taken out and placed in different molds and aligned and equipped with labels before polymerization. And the results were all very good. So ultrastructure was preserved very well, and yeah, the samples were very useful. So here I know we have some groups here at Northwestern that use elaborate staining protocols for serial block phase imaging or in general for volume electron microscopy. And here we see, for example, one of these protocols. So we have several staining steps, several incubations with osmium, with uranyl acetate, with lead aspartate. And so these protocols take really a very long time. Here, all in all, about 40 hours before polymerization. And it's clear that doing this in an automated way would be a very big advantage. And here we also can see some sealed capsules. So we have plastic seals for aqueous solutions, and we have those aluminum heat seals for solutions that contain solvents that easily evaporate. So here's an example for these processed samples, so manually processed with this extensive protocol. And here on the C is an automated processed sample. And the quality of all samples is absolutely comparable. So it means it's to automate this process leads to really very useful samples and saves a lot of time. And here again, we can see that the whole block is well preserved after automated processing, and we can easily reconstruct synapses, mitochondria, and myelin sheath in a given sample. And as I said, the whole block shows very good penetration of all agents. So here we see a comparison between 2 protocols, the manual protocol, and on the right in green, the automated protocol. And we can again see that the automated protocol is way more effective. So here, for example, for the buffer rinses, we have 6 exchanges of solution for 30 minutes. So that means 6 times 5 minutes. Here we have 90 exchanges in 3 minutes in 3 different solution trays. And the same is true for all other steps here. 5 times 5 minutes in water. Here we have already 450 times of exchange in 5 different wells of water. So here we are below or around 10. We have around 10 exchanges, and here we have hundreds of exchanges in a shorter amount of time. So, and when we look at the result, we can see that the automated processed samples look at least as good as the manually processed ones, maybe even better. And when we look at the time, we see we have 2 full days of work for the manual protocol. And here we have 1 hour of work, which I believe is a bit short. And let's make that 2 or 3 hours, depending on the experience level that a user has, but it's still a huge difference between manual and automated processing. So, and there's also this case where we can use this clamping mechanism for the capsules, for sample capsules. And we can also, for example, include fiduciary markers that allow us better to estimate the position where we are with sectioning. This has been done here. And here, again, the whole capsule is polymerized, and we can see nicely our marker. In this case, sections were used for SEM. And it was also possible to auto-segment very well labeled or very well stained exons in that sample. So this is just, another possibility how to align samples in those capsules. One advantage of these capsules is also sometimes we have samples that contain gas or air bubbles, and those samples always would swim up on the surface of solution, and we would never have a real good exchange. And in this case, we also can use capsules either by stacking or by using the screen and keep those samples all the time submerged. So this might give you the ideas with your samples. It need not to be biological samples, but any sample that needs to go through incubations with solutions can be used, I guess. So here again, when we look at the time effort and compare the different methods, regardless if it's preparation for TEM, if it's for TEM immuno gold labeling, if it's for volume EM or for SEM, in every single case, we can see that the automated processor requires always way less processing time than all the other methods. Okay. So again, the benefits for the user with using automated sample prep are the main benefits, of course, only there are way more, is high time efficiency and scalability. So users have more time for other tasks and they have more samples ready in less time. We also have improved consistency because the sample prep is absolutely repeatable, it's standardized, and that means we minimize the variability between samples. We also have reduced errors because manual handling errors are eliminated, and that means our results have a maximum reliability. And last but not least, we have increased safety because the APS is connected to an exhaust and manual handling of chemicals is reduced. It's only necessary when those plates are being prepared. With this, I want to show just one more thing. We have a new biocrio development here. We developed a new protocol for cell monolayers on cover slips, and so far, the first experiments looked very good. And as we can see here on a low-mag image and on a higher zoomed-in image, that shows very well preserved cristae in mitochondria in this thick section of a cell. So with this, I hope you got a good impression of what automated sample processing can do, and I'm ready for your questions. Of course, you can always reach me via email, or you can visit me. You know that I live in Hogan 5156. Or you can reach me by phone, of course.